Okay. Just for everyone who's joined, make sure you follow the safeguarding requirements. So microphones and cameras should be turned off, and if you're under 18, please inform parent or guardian that you're attending the session, and if you have any questions, type in the chat. So as I was saying, today we're going to be looking at investigating diversity, and the learning objectives are to be able to explain the different ways of comparing genetic diversity, and to explain how data could be collected, processed, and analysed when investigating variation within a species in a quantitative manner. So in terms of what this specification actually wants you to know, genetic diversity within or between species can be made by comparing the frequency of measurable or observable characteristics, the base sequence of DNA, the base sequence of mRNA, and the amino acid sequence of the proteins encoded by DNA and mRNA. So you should be able to interpret data relating to similarities and differences in the base sequences of DNA and in the amino acid sequences of proteins to suggest relationships between different organisms within a species and between species, and to appreciate that genetic technology has caused a change in the methods of investigating genetic diversity. Inferring DNA differences from measurable or observable characteristics has been replaced by direct investigation of DNA sequences, and the knowledge of gene technologies will not be tested because that's something that you cover right at the end of the specification in Module 8. So to begin, I think the key sort of introduction to this section is phylogenetics. And this comes from the Greek word philo, which means tribe or race, and gene means or relates to the word genesis, meaning how something develops. Okay, and just so you know, things that I've determined important and worth putting in your notes, I've highlighted in yellow. So phylogeny is the grouping of organisms based on their evolutionary relationships using common ancestry. And phylogenetics is the study of how the evolutionary history of groups of organisms to determine how closely related they are to one another. So I think many of you would have seen something like this before, and it's called a phylogenetic tree. And essentially, it's a diagram showing the phylogeny or evolutionary history or relationship of groups of organisms. So they're branch showing which groups have evolved from a common ancestor. Okay, so the nodes, or branching points, show shared or common ancestors, and all organisms have evolved from a single shared common ancestor, which we refer to as the last universal common ancestor, or LUCA. And organisms without many branches evolved early and are the most different. So when an organism evolves, it's not only the internal and external features that change, but also the molecules that are made, so the actual proteins and enzymes, for example. And DNA determines the proteins of the organism, and proteins determine the features. So it follows that changes in the features of a species are due to changes in its DNA. So if we compare the genetic diversity between species, then we can find out how closely related species are to one another. So traditionally, we've used something called comparison of observable characteristics to find out how closely related species are. And this essentially just is about physical observation. And the basis of this is that the characteristic is determined by genes, and the variety depends on the alleles of the gene. But of course, there are limitations because a large number of characteristics are coded for by multiple genes, and characteristics can also be modified by the environment. So height is a product of both multiple genes and your environment, and it's not precise if only we look at one characteristics. So if we to look at the number of legs, many animals have 4 legs. So therefore, inferring DNA differences is replaced by actually looking at the DNA sequences themselves. So this is possible through gene technology. And further examples of characteristics that could be observed include color, patterns on fur, scales, feathers, habitat, and the presence of hair or wings or fins. So I'm not sure if any of you know this, but whales are actually genetically closer to snakes than sharks. And this is shown on the phylogenetic tree. So if you look at the branching points in yellow or orange, you can see how whales have a more recent ancestor to snakes compared to sharks. Because you need to go further back on the phylogenetic tree to find their common ancestor. And the reason for this, which you don't specifically need to know, is because of convergent evolution, where it's due to the environment that organisms can have similar features, i.e. because they're in the sea, for example, and not because of their actual DNA. So in terms of gene technology, you can look at DNA-based sequences. So this means the exact order of the nucleotides. And DNA sequencing is now routinely done by automatic machines, and the data produced analyzed by computers. So each nucleotide base can be tagged with a different colored fluorescent dye. So, for example, adenine could be green, thymine could be red, cytosine could be blue, and guanine could be yellow. And this essentially produces a series of colored bands, which you can see below. And each one represents one of the 4 bases. And then analyzing these patterns allows us to compare one species with another, or one individual with another of the same species, to determine how diverse they are. And we can use these to determine the evolutionary relationships, as I've said before. So when one species gives rise to another species during evolution, then the DNA will actually be very similar to that of the species that gave rise to it. And due to mutations, the sequences of nucleotide bases will change. So therefore, new species will accumulate more differences in DNA. And then we'd expect species that are more closely related to show more similarity in their DNA-based sequences than those that are more distantly related. And just to remind you, when I talk about DNA mutations, I mean mutations such as deletion mutations, where you have certain nucleotide bases being deleted, or insertion mutations, where you have DNA bases being inserted, or even substitution mutations, where, for example, you could replace a adenine with a cytosine, for example. And this would all lead to an altered DNA sequence. Next is comparison of the base sequence of mRNA. So mRNA is coded for by DNA, and the base sequences are complementary to the strand of DNA from which they're made. So by comparing the base sequence of mRNA, we're inadvertently measuring DNA diversity and therefore genetic diversity. And the reason why this could be somewhat easier is because we can have a loss of mRNA in the cytoplasm, and these are easier to isolate compared to the DNA. And the mRNA doesn't have an intron, so an intron is a non-coding region of DNA. And just as a reminder, I've put the diagram in the bottom right, which shows how DNA and RNA are related. So the next thing is comparison of amino acid sequences. And like before, the sequence of amino acids is determined by mRNA, which is determined by DNA. And genetic diversity can be measured by comparing the amino acid sequences of their proteins. The degree of similarity in the amino acid sequence, i.e. number of similarities or differences in the amino acids, will reflect how closely related the 2 species are. So if you see on the images, we have 7 amino acids in 6 different species, and then the table on the right shows the number of differences and number of similarities. I would advise you to only focus on either the bottom left or the top right, because they both show the same information but the inverse. So if you just take a look at the top right, for example, you can see how species 6 and 1 only have 1 difference. Specifically, the 5th amino acid is valine in species 6, while it's glutamate in species 1, for example. So therefore we can say that 1 and 6 are somewhat closely related and they have a more recent common ancestor. While if you were to compare 1 and 2, you have methionine changed to alanine and valine changed to methionine as well. So they have perhaps a more distant common ancestor. So here's an example from the textbook, just to illustrate how you can, if you were to get this in an exam, how to find out which species are more closely related. So we have a human, gibbon, gorilla, orangutan, chimpanzee, and lemur, and then the different amino acids, so each represented by a letter. And then if it's a different from the reference, so if we're using humans as a reference, then it's in red. If it's the same as the human amino acid in that position, then it's in black. So the first step is to find the number of differences, which is 6, 2, 4, 0, and 5, with the humans as a reference. Next, you can use this to find out what the phylogenetic tree, because those with fewer differences would have a more recent common ancestor. So for example, we know that chimps and humans have no differences, so they're probably the same section of the phylogenetic tree. Gorillas have 2, so it's further away, and then orangutans have 4 differences, and then lemurs and gibbons have the fewest number of similarities with the human amino acids. And that's how you would construct your phylogenetic tree if asked in an exam. Next, we're going to take a look at quantitative investigations of variation. So the specification says that this would involve collecting data from random samples, calculating a mean value of the collected data and the standard deviation of that mean, interpreting mean values and their standard deviations. And just to let you know, you won't need to calculate your standard deviations in your exams. This is more for your write-ups to achieve your required practical requirements. So the first thing is to know is that it's very, very hard to measure or investigate the properties of every single organism in a species of interest. So if you're looking at a forest, it's hard to find the height of every single tree. This is why we use random sampling. Now, sampling involves taking measurements of individuals selected from the population of organisms that is being investigated. So if they're representative of the population as a whole, then we can say that the measurements can be relied upon and our results and conclusions are probably reliable as well. But in reality, it's hard for our sample to be perfectly representative because there could be bias. So for example, the investigator might make unrepresentative choices, either deliberately or unwittingly. So one good example is whether if you were investigating something, you'd be likely to take samples of buttercups from a muddy area and a dry one, or if you'd avoid areas covered in cow dung or had lots of nettles. And even if there was no sampling bias, there is always an aspect of chance where the individuals chosen aren't representative. So for example, the 50 buttercup plants selected might just happen to be the 50 tallest in the population. Even though this is very, very unlikely, it could still happen. And therefore, our sample might not be perfectly representative of the entire population. So, something that is very, very high yield and comes up nearly every year, in my opinion, is performing sampling. And there's a list of points which should be in your notes, just so that you can get full marks if you get these sampling questions in the 2026 exams. So if you were asked to outline how to perform unbiased, representative random sampling with a quadrat, you could mark a grid in the area to be sampled using perpendicularly placed measuring tapes to act as axes. And then you can use a random number generator. So, for example, you can use the ranint function on your calculator to provide coordinates on a grid. And then you can place your quadrat at coordinates and count species or organisms within a species. And then you could, for example, use an identification key to improve the reliability of your count. So, for example, if you're looking at flowers and in that area there happened to be two different species of flowers which looked very similar, you could use a identification key to make sure that you are actually counting the flower that you're interested in. Then you can, for example, if you're looking at plants, you can determine abundance, if that's what you're investigating, via percentage frequency or percentage cover. And you should ensure there's a standardized sampling procedure. So if you're counting flowers, you might decide to only count flowers that are completely in the quadrat, as opposed to half in and half out. And you should also use methods to prevent recounting. So, for example, if you mark a fish, for example, with a non-toxic dye that doesn't affect its survival rate, and it doesn't rub off, then if you were to capture that organism again, then you could ensure that you're not recounting the same organism again. And then you could repeat many times. A number that I always go for is 50. Realistically and practically, it's probably a bit too high. I think in exams it's above 15 that they like to accept, but it doesn't hurt to go way higher just to be sure. And then you can calculate a mean, which I'll mention later. If necessary, sample at different times or seasons, and where applicable, ensure the sampling is ethical and the organism is not harmed, and it's released into its original habitat. Although the quadrat method is typically for non-moving organisms. If you were to use some like a capture-recapture technique, this last point is particularly applicable. Next is how to perform unbiased sampling with a belt transect. The image on the bottom right shows you the difference between a line transect and a belt transect. So a line transect is where you record organisms found at the regular sample points. So, for example, you put 2 points and some line between them, and then you see how many, for example, flowers are under the wire. And then a belt transect is where you put a quadrat at regular sample points, and then the abundance of the organisms within each is recorded. And the reason why transects are used is if there is some kind of biotic or abiotic factor that is changing with distance from a certain point. And the method for this is to set up your transect along the area that you're thinking of sampling. Then you're placing your quadrat at regular intervals and counting the species or organisms within the species. Use an identification key again to improve the reliability of the count. Determine your abundance, so percentage frequency or percentage cover. Repeat many times, so once again, maybe 50 times. Calculate a mean, and then if necessary, sample at different times or seasons. In the bottom left, they've got an example of a hillside where you can clearly see, for example, flowers have much higher density at the bottom compared to the top, maybe, and you've got a belt transect, and you're sampling along the belt transect down the hillside. Next, we're thinking of key sort of statistical techniques, so calculating mean, median, mode, and range. The mean of a data set is calculated by adding all of the values together and dividing by the total number of values. So the mean of, for example, the numbers 3, 5, and 1 would be 3 plus 5 plus 1 divided by 3, which would be 9 divided by 3. So the mean of those three numbers would be 3. The median value is calculated by arranging all of the values in numerical order, and the middle value is the median. In maths, you're taught the formula n plus 1 divided by 2, where n is the number of values in the data set. So for example, if you had 3, 5, and 1, once again, as your values, you have to order them numerically into 1, 3, and 5, and using our formula, n plus 1 divided by 2 would be 3 plus 1 divided by 2, which would be the second value, which would be 3. Next, the mode of a data set, it can be calculated by finding the result or the value that occurs the most, and then the range is the difference between the highest and lowest results. So once again, with my example of 3, 5, and 1, the range would be 5 minus 1, which is 4. Next, we're going to think about why means, the advantages and disadvantages of a mean. So the mean of a sample of data provides an average value, so it can be used for comparison. However, it doesn't provide any information about the range of values, for example. And this means that it's susceptible to outliers. So once again, if you have 3, 5, 1, and then 35, the 35 will heavily skew the actual mean value that we get, even if it may be a perfectly valid result for whatever reason. So, a glimpse was provided on the previous slide, but we're going to take a look at the normal distribution, which is something that is covered in maths, and not all of you may be taking maths. So a normal distribution occurs when you get some kind of data set and you get a bell-shaped curve when the data is symmetrical about the mean. And what this means is that half the values are less than the mean and half are above the mean. And most values are close to the mean, and the values of the extremes are low. So just remember that the mean is the average of all the values, so the mean would be in the middle of the normal distribution. The larger the sample, the more likely it is that the data will follow normal distribution, and the closer that the mean will be to the actual mean of the population. So the larger your sample, the more representative it will be of the whole population. And this is something that I'll show you visually slightly later on. So you can see on the bottom right the normal distribution curve, where the mode is equal to the median, which is equal to the mean, and they're in the middle. And this bell shape is typical of a continuous variation. So for example, height in humans, most people would be in the middle, with 50% being higher than the mean and 50% would be lower than the mean. And the graph would be symmetrical, therefore, around the central average height. Occasionally we could get something called skew, which was shown on the previous slide, and this is where you have the curve being slightly shifted to one side. So the example as shown is where you've got the curve being slightly shifted towards the right, and the mode is higher than the mean, which is higher than the median. And this is skew. Next, another topic that is covered in A-level maths is standard deviation. And just as I've said before, they don't need you to calculate standard deviation in your exams, but it's something that you might need to do for your practical write-ups. And you might also need to interpret standard deviation values if given, for example, in a table. So standard deviation is essentially a measure of spread. It tells you how spread out the data is. And the greater the standard deviation, the greater the spread. Okay, so once again, if you were to get given data in a table and you see that there's an overlap in standard deviation, then it means that the differences aren't significant. But if there's no overlap, there's a significant difference between the results. And because standard deviation is a measure of spread, if you have a large standard deviation, then you have a large amount of variation. So if you were to have a normally distributed data set with no skew, the middle 68% would be of the data would be on one standard deviation on either side of the mean, and then 95% of the data would be within two standard deviations on either side of the mean. And this is seen in the yellow curve, in the green curve on the bottom right. You can also see how having different standard deviations also impacts how the curve looks. So for example, on the left graph, you can see how a standard deviation of 3 would mean that you've got a curve with a higher peak compared to, for example, a standard deviation of 9, where you've got a much broader range that the data can take, much greater spread of data. And as I've said, you don't need to calculate standard deviation. This is only for your practical write-ups. But just so that you're aware, the formula for standard deviation, and I will go through a worked example with you, is the square root of the sum of a value minus the mean squared divided by n minus 1, where this sort of inverted E means sigma, which is the sum of x is the measured value from the sample, and then x with the line on top, or x bar, for example, as you may call it, is the mean value, and then n is the total number of values in the sample. So I've got this worked example from the textbook. Let's say you were to get 4, 1, 2, 3, 5, and 0. First step is to calculate the mean value, so x bar, and this is 4 plus 1 plus 2 plus 3 plus 5 plus 0, which is 15, and you have 6 values, so you divide your 15 by 6 to get 2.5, and then what you would do is subtract the mean value from each of the measured values. So this is what the x minus x with the line on top represents. So you would do 4 minus 2.5, 1 minus 2.5, 3 minus 2.5, 5 minus 2.5, and 0 minus 2.5. And you'd get 1.5 minus 1.5 minus a half plus 2.5 and minus 2.5. And then you would square them so that all of these numbers are positive, because you can't have a negative square root, or the result of square rooting a negative number leads to an imaginary result, and you can't have an imaginary standard deviation. So we square all of these numbers to get 2.25, 2.25, 0.25, 0.25, 6.25, and 6.25. And then you get to the sum of all of these numbers, which is 17.5, and then you divide by n minus 1. So n is the number of values, so n minus 1 would be 5. So you do 17.5 divided by 5, which is 3.5. And then as all of these have been, and then the final step after you've done all of this is to square root your 3.5, which gets you 1.87. So the standard deviation of the sample of the values 4, 1, 2, 3, 5, and 0 is 1.87. So 68%, you could say, if you had a very large data set, lie 1.87 away from the mean. Or in this case, 68% of these 5 numbers is 1.87 above or below 2.5, because that's your mean. Okay, I said before that we would take a look at the importance of having a large sample. So let's assume we're sampling from a normally distributed population with the following properties. So the mean is 50. So let's say we know the actual mean and the standard deviation, and we're testing the accuracy of our sampling. So let's say the actual mean is 50 and the standard deviation is 10, and you get someone else to go out into the field and take a sample and tell you their results, and they decide to take 4 samples because they're feeling tired and they're hungry and they're looking forward to their lunch, and they get back with these 4 dark blue bars. So the red curve is actually what we would expect if we were to take a very large sample size, as we'll see later on. But with the person taking 4 samples, you get a mean of 57.4 and a deviation of 6, and this is not equal to the actual mean and standard deviation of 50 and 10. So as you can see, this light blue line, which should have been in the middle at the peak of this red curve, is shifted to the right, and this is why we need to take large samples. Next, if we were to take 1,000, because we're feeling very patient and diligent, then we'd get the following results, and you can see how we follow the curve to a much better extent. So with a mean of 50 and a deviation, with a mean of 50.4438 and a standard deviation of 9.7803 with our sampling, that's very close to the actual real values of 50 as our mean and standard deviation of 10. And this is why we need to take a large enough sample size so we get representative, reliable results. Okay. That is a whistle-stop tour of 3.4.7. I would say that it's not a lot, but something such as sampling is very, very high yield, and it's very likely to come up because it tests a lot of the practical skills, because it comes under RP12. It also covers the content, and they can also bring some maths in, so they get to do a lot of things under the umbrella of sampling, which we covered in just now. But to recap everything we've done, we know that genetic diversity within or between species can be made by comparing the frequency of measurable or observable characteristics, and we can also compare the base sequence of DNA, the base sequence of mRNA, and an amino acid sequence of the proteins encoded by DNA and mRNA. And you should be able to interpret data relating to similarities and differences in the base sequence of DNA and in the amino acid sequences of proteins to suggest relationships between different organisms within a species and between species, and appreciate that gene technology has caused a change in the methods of investigating genetic diversity inferring DNA differences from measurable or observable characteristics has been replaced by direct investigation of DNA sequences, and quantitative investigations of variation involves collecting data from random samples, calculating a mean value from the data and the standard deviation, and interpreting mean values and their standard deviations. And I've got some really cool questions just to test your understanding. So there are 21 questions on the slide. Maybe you can take 5 minutes in your head thinking about what you'd write. Or alternatively, you could do this on paper if you wanted to. If you do have any questions, do put it in the chat and I will take a look at them. Before you leave, there is a feedback form which I will put into the chat. So if you could do that, I'll be greatly appreciated. But yeah, for now, you can take a look at these really cool questions and I'll show you the answers in 5 to 10 minutes. So I've put the feedback form in the chat, and if you could fill that out, it'd be greatly appreciated. And once again, if you do have any questions, do put it in the chat and I will do my best to answer them. I'll give you maybe 3 more minutes before I go through the answers. Okay, I'll give you one more minute before I go through the answers. Okay, time to go through the answers. So, what is sampling? Sampling is where you take measurements of a limited number of organisms of an area. It's important because it's impossible to count or measure all of the organisms in a particular area. It's used to estimate the number of organisms or the particular characteristics of those organisms without counting all of them. And it's carried out using a random number generator that you're not involved in choosing. You use two tape meshes as a grid, and the random numbers generate the coordinates, and samples are taken at the coordinates. Reliability is a measure of how representative of the organisms present in the habitat the sampling is, and how accurate the sampling is. Sampling bias and chance can reduce the reliability of the sampling being done, and sampling bias is where you accidentally or deliberately choose a particular area to sample, and this is reduced by random sampling. You cannot remove chance, but you can reduce its effect by having a large sample size, which I showed you visually in previously using the graphs. The most valid way of sampling an area is random sampling with quadrats. If you're looking at organisms that don't move, you can sample with quadrats by placing a large number of them, such as, for example, 50, randomly using grid coordinates and a random number generator, and then calculating a mean and standard deviation. A mean is calculated by adding the individual results, then dividing by the number of repeats. A normal distribution is data that creates like a bell curve when plotted. The characteristics is where it's symmetrical about the mean, so 50% of values are less than the mean, 50% of values are greater than the mean, and most values are close to the mean, and the values at the extremes are low. If there's a high standard deviation, there's a lot of variation in the data and the results. If there's a low standard deviation, there's a small amount of variation. Okay, next. You can measure genetic diversity and phylogeny by looking at the frequency of measurable or observable characteristics, such as the similarities in DNA base sequence, mRNA base sequence, and similarities in amino acid sequence. Next, you can, to explain how comparative biochemistry provides the evidence of genetic diversity. If you're to take a study of similarities and differences in proteins and DNA of an organism, mutations that in these molecules can help identify genetic diversity, and species that are closely related have the most similar DNA and proteins. If you're asked how DNA sequencing is used to determine evolutionary relationships, you can look at the difference in the number of mutations between species and calculate the average mutation rate, and this can be used to determine when the two species have diverged. Next question was, how was DNA sequencing changed how species are identified? And traditionally, species identification was done by observation of anatomical and the physiological features, for example, number of legs, the color of the fur. But in DNA sequencing, genome similarities are examined, and then comparisons are made to the standard species genome. And this is more accurate, but it's harder to carry out in the field. And the importance of sampling in measuring the biodiversity of the habitat is, as I've said before, it's difficult to count every individual. The sample provides an estimate, and the sample should be representative of the whole area. Finally, this is the last question. It's, I'd say, a pretty hard question to get full marks in. So hemoglobins are chemically similar molecules found in many different species. Differences in the primary structure of hemoglobin molecules can provide evidence of phylogenetic or evolutionary relationships between species. Explain how for 5 marks. So this could be like one of the subparts in the final question that you can get in A-level paper 1. And I will give you until 55 to answer. You can bullet point if you want to what you would say for this question if it happened to appear in your exam. One more minute before I go through the answers. Okay, so hemoglobins are chemically similar molecules found in many different species.